right, welcome to the review of lab number 9. So we'll just go over these answers really quickly, and then I have posted this key in D2L as well, so you can check it out there if you'd like. So in the pre-lab, we needed to watch this video on how to use a micropipetter. So then as far as these questions are concerned, when it comes to the first step to transfer a sample to a new container, which is after you put the tip on the micropipette, first thing you have to do is depress the plunger to the first stop. Once you press it to the first stop, then you put it into the sample and release to draw the sample up into that pipette tip. You press and hold the plunger at the second stop when you are delivering the sample to its new container. So once you have drawn up the liquid into the tip and you put that tip into the new container, you press to the first stop and the second stop to fully release all of the liquid into the new container. If your micropipetter is blowing bubbles in your sample, that means that you didn't press and hold the plunger before placing it into the sample, right? Just like if you have a straw in a glass and you blow liquid into that straw, the liquid kind of bubbles up in the glass. It's the same thing, right? If you put the pipette tip into your sample and then press down the plunger, you're expelling air into that sample. So that's where you're getting those bubbles from. And then if you release the plunger too quickly, you get a few things happening. You get an air bubble that forms, your measurement's incorrect, and it's possible to damage the micropipetter. So there's a few different answers there for question number 4. Okay, when it comes to the steps of PCR, polymerase chain reaction, the purpose of the primers is to attach specific DNA sequences of interest rather than the whole strand. We don't want to copy the whole thing, we just want specific pieces. So that's what these primers are for. The temperature that DNA polymerase starts to work is 72 degrees Celsius. Remember that in PCR, we're cycling that temperature to higher and lower temperatures depending on what is happening. So when DNA polymerase is working, that's at 72 degrees Celsius. And then once we've done 30 cycles of this PCR, we have over 1 billion fragments that contain our target sequence. So we have really increased the amount of DNA we have in that sample. So after 30 cycles, we get over 1 billion fragments. So then there was another video to watch on the steps of gel electrophoresis. So the bands that appear in the gel, those are groups of stained DNA strands of varying lengths. And then which fragment moves the farthest? So that's going to be the smallest segment. So BP means base pairs. So we have the smallest number of base pairs here with 1,000. That's the one that's going to move the furthest. So then this question here, I'm not going to go over this right now. We'll talk about this when we get down to part number 2. I just copied and pasted those answers up from that part of the lab. So we'll get there in just a second. All right, so in the actual lab activity, we had to go through this simulation of gel electrophoresis. So when you went through that, you could answer these questions. So the charge associated with DNA is negative. So DNA has a negative charge on it. The purpose of the wells in the gel, so it's these indentations that allow the samples to travel through the gel, right? We need to get our DNA into the gel, not just sitting on top of the gel. So the wells allow us to put the sample into the gel. As the DNA molecules travel through the gel, they move away from the well towards which end? They move towards the positive end. DNA molecules are negatively charged. Negative attracts positive. So they are going to, when that charge is applied, they are going to move away from the negative and towards the positive charge. Explain why DNA molecules of different sizes travel different distances. So that gel that we have has pores in it, and these holes, it's really easy for the smaller pieces to pass through these holes and travel around really easily, but those large molecules get stuck, so they can't go very far. So that's why the smaller pieces move further down the gel. Explain the purpose of using a DNA ladder. So the DNA ladder is known sizes of DNA. So if we put that ladder in there, we know what those bands are, how big those pieces of DNA are. So then if we compare that to our unknowns, we can get an estimated size of those unknown fragments, those length of base pairs. So in our lab, we would be analyzing linear DNA. There's some problems analyzing circular DNA with gel electrophoresis. They can have various conformations that impact how they travel. So they can kind of twist upon themselves, which makes it a little bit difficult to understand the number of base pairs. Depending on how that circular piece of DNA is oriented, the same number of base pairs could show up as different bands on the gel. And then explain the purpose of the loading dye. So it not only causes the sample to sink, but also allows us to see that the sample is in the gel. It allows us to see that the DNA is running in the correct direction, and it allows us to see how far down the samples have gone in the gel. Okay, so part number 2, we are diagnosing a genetic disease here. We're playing the role of technicians in a hospital. So to give us some background here, we've got Mary and John Smith that have 3 children, Daniel, Alice, and Michael. Daniel was having difficulty climbing the stairs and was really tired, so he had some tests done, and he has this type of muscular dystrophy, DMD, right here. So it's a genetic disease that results from damage to the gene on the X chromosome. So this is an X-linked trait here. DMD is more common among males. They inherit the defective copy of the gene from their mother, right? These X-linked recessive disorders are always more common in males because with females, a lot of times they're just carrying that disorder with one copy of that recessive trait, and they have a dominant copy to overpower it. But with the males, they only have one copy of that X chromosome. So if it's defective, then they are going to have that disorder. Okay, so the Smiths are concerned about their remaining children, and they want to know their chances. Mr. Smith does not have DMD, but Mrs. Smith must be a carrier, right? That's how Michael got that disorder. Alice might not have DMD, but she could be a carrier. And Mary just found out that she's pregnant, and so she's concerned about her unborn child being affected or being a carrier. All right, so here's our Punnett square. So we know that Mrs. Smith is a carrier, so she has one X that is the dominant allele and one X that is the recessive allele. She's carrying this recessive allele. And then Mr. Smith does not have DMD, so he has this dominant allele and a Y. So when we do the Punnett square here, we can see that we have a possibility of a female not being a carrier at all. We have a possibility of a female being a carrier. We have a possibility of a healthy male, and we have the possibility of a male with DMD. Okay, so we've got all sorts of possibilities there in that Punnett square. Okay, so we are basically going to be taking blood samples of all of these members of the family and running a gel to sort of look at what types of alleles they have. So if you go down to this paragraph, you can see that Mary has 2 copies of the DMD gene. One is normal and one is defective, right? One is the dominant one is the recessive one. Because he is male, John only has 1 copy of this gene because he only has one X chromosome, and it's a normal allele. Daniel only has 1 copy of the gene, and it's the defective allele. So the fact that we know the genetic makeup of these individuals, we can interpret the genetics of the other individuals of the family. So Daniel's genotype here, he has 1 copy of the X chromosome, and it is the defective allele, and then he has 1 Y. Okay, so we have our gel here. We've got band number 1 and then band number 2. Okay, well A is mom, well B is dad, C is Daniel, and then we have D, E, and F as our other children that we don't know their genotypes. Okay, so if we look at A, this is mom. So mom has 2 copies of the allele. She has 1 normal and 1 recessive. If we looked back up in this paragraph, where did it go? Yes, the defective allele has a piece of DNA deleted from it. Okay, it has a piece of DNA that's missing, so it has fewer base pairs. Because it has fewer base pairs, it's going to travel down the gel further. So mom has a normal allele up here, and then her defective allele has traveled further in the gel because it is a smaller piece of DNA. All right, so she has 2 copies of that allele, and that's her genotype there. 1 regular, 1 that's defective. Dad is over here in this well. He just has 1 copy of the allele, and it's normal. So he has this genotype here. Daniel is in this 3rd well. He only has 1 copy of the allele, and it's defective. You can see that one in this allele that's traveled further. So this one is the daughter Alice. So she, like her mom, is going to have 2 copies of the alleles. she has one band up here and one band down here. So she has one copy of the regular allele and one defective allele. So that's her genotype right here. She is a carrier. This son, Michael, only has one copy of the allele because he only has one X chromosome, and that is going to be up here in band number 1, which is just like his dad. So he has a regular allele. He is a healthy male. This is his genotype down here, a regular D or a regular X chromosome, and then the Y chromosome. And then this is our fetus, our unborn child. We can see that there are 2 bands. So there are 2 copies of the X chromosome, 2 copies of that allele. So this is going to be a female, and they are going to be a carrier, right? This looks just like the daughter Alice over here, and it looks just like the mom. So there are 2 copies of this allele. One is the regular, and then one is the defective. Okay, so that's how we fill in that chart there. So are the DNA molecules positively or negatively charged? We know they're negatively charged. They traveled towards the positive end of that gel. Which allele moved further in the gel? The defective allele moved further because it's smaller. It has a piece that's been deleted, so it can move further in that gel. Is Alice a carrier? Yes, she is a carrier because she not only has a copy of the regular allele, but she has a copy of the defective allele as well. So she's not impacted by DMD, but she could pass it on to her offspring. That's what it means to be a carrier. All right, does Michael have DMD? He does not. He only has one copy of the allele, and it's a regular allele. He doesn't have any defective alleles. What about the Smith's unborn child? That unborn child is a female because there are 2 copies of the allele. That's how we know that it's female. If it was a male, it would only have one copy of that allele because it would only have one X chromosome. Does the fetus have DMD? It is a carrier for DMD. It has 2 copies of the allele, one regular and one defective. So that's how we know that this fetus is a carrier. Can a male be a carrier for DMD? No, it cannot. A male has 2 options. They can be healthy or they can have DMD. They only have one copy of the allele, so they can't carry that defective allele and not have the expression of that disorder. Okay, so moving on to our last section here, part number 4. So we gave a little example here of how plants have been used in forensic investigations, basically comparing plants at a crime scene to samples that were taken from a suspect. So in this example here, we're talking about seed pods where the parent plant and the seed pods are genetically identical. So if we're comparing 2 different samples, we would expect them to be exactly the same. So these are the results here. We have the plants at the crime scene and then 5 different samples. Which letter corresponds to the largest DNA fragment? So letter A corresponds to the largest fragment because it has moved the smallest distance, right? The largest fragments are going to be up here. They don't travel as far. The smallest fragments are going to be down here because they do travel further. Okay, so our large samples up here, smaller fragments down here. And then which sample number came from the seed pods found in the suspect's truck? So here we're looking at the suspect was convicted, right? It says it right here. So we want the sample from our suspect's truck to be the same as the plant from the crime scene. So if we're looking at these bands over here, we want a sample that has all of those same bands. So sample 1 is close, but down here it looks a little different. Sample 2 is missing these bands here. Sample 3 is missing this band as well as some bands down there. Sample 4, that one is identical. Sample 5 is not. So sample 4 is identical to the plant at the crime scene, so that is the one that is going to be from the suspect's truck. Okay, then the last part here, we're talking about paternity tests. So we have STR, short tandem repeat profiles, that can be used to identify a child's parents. A child gets half the DNA from mom, half the DNA from dad. So how can a child's profile have bands of DNA that don't match the mother? Those are the ones that come from the father, right? It gets half from mom, half from dad. So if it doesn't match mom, it has to match dad. So we've got a local buffalo farm. We have an albino calf named Snowball who was born, the first albino buffalo to be born in this herd. So the allele for albinism is recessive to the allele for normal coloration. Snowball's mother, Bessie, has normal pigmentation. And for future breeding purposes, the owner wants to know who of his normal non-albino bulls are the father. So we've got Scout, Chief, and Thunder. So we've got this genetics company that's going to determine who is the father of Snowball. So this is our gel results here. This is our control. It's just showing the number of bases. This is our DNA ladder, so known number of bases. We've got Snowball here, we've got Bessie here, and then we have the 3 fathers over there. All right, so which of the bulls is Snowball's father? So the father is Thunder, and these next few questions are going to help us determine how we figured that out. So let's talk about Scout first. So that's question number 2 down here. So if we look at these bands, this band matches with Bessie, so we don't need to worry about where this one came from. This band does not match Bessie, so we need to figure out which father has this band. Well, Scout, Chief, and Thunder all have that band, so that's not really helpful. This band, we see that in Bessie, so we don't need to know where that one came from. This band right here, Bessie doesn't have that one. Chief and Thunder have that one, but Scout doesn't. So if Scout doesn't have it and Bessie doesn't have it, but Snowball does, Scout can't be the father. So that's what this one is saying down here. Snowball has a DNA sequence that's approximately 8.5 kilobases that is not present in Bessie, meaning it must come from the father. Scout does not have a segment of this length, so he is not the father. So we can exclude Scout from this test here. So then if we keep going, we've got another section here that corresponds to Bessie. This band corresponds to Bessie. This band corresponds to Bessie. This one also corresponds to Bessie. So none of those are going to be helpful for helping us figure out who the father is. But then this band down here, Bessie doesn't have that. Scout doesn't have that, but we already ruled him out. Chief also doesn't have this band. So that's what we're saying down here. Snowball has a DNA sequence that's approximately 2 kilobases. That's not present in Bessie, meaning it must come from the father. Chief does not have a segment of this length and is therefore excluded. So now Chief is out. So that only leaves us with Thunder. And if we look back to these 2 segments that we talked about before, this 8.5 kilobases and 2 kilobases that are not present in Bessie, meaning they must come from the father. Thunder has both of these sequences and is therefore included. So this one down here, Thunder has that one. This one up here, Thunder has that one. So Thunder is our father. All right, so if you have any questions about this lab, let me know. I haven't gone in and graded the ones that have been submitted yet, but you can expect to see those grades pretty soon. So again, this key will be posted in D2L as well. So if you want to check out the answers, feel free to take a look at that. And I will see you guys on Monday for the live lecture.